CRISPR-Cas9 is a technology that enables gene editing by removing, adding, or altering a DNA sequence. Initially discovered in the immunological defense system of bacteria, it provides astounding accuracy and can be performed within live cells themselves. Firstly, an RNA scaffold containing guide RNA with bases complementary to that of the DNA strand binds to a specific DNA sequence. The Cas9 enzyme then follows this guide RNA and cuts across both DNA strands. Cells then recognize this damaged DNA and try to repair it, and at this stage scientists can insert a mutant copy of the DNA that can pair up with the cut ends of the DNA and recombine the strand.
The modular component of the CRISPR-Cas9 system, the guide RNA, the part that we change, is only 20 base pairs long and very easy to tailor. You only need one CRISPR-Cas9 system to make the double-strand incision, whereas in the two previous methods you need at least 2 talons or zinc fingers to make the incision on both sides of the DNA strand. CRISPR is just a far more elegant system.
Here is a brief timeline of major CRISPR events. In 2002, the term CRISPR was first coined. This stands for Clustered Regularly Interspaced Short Palindromic Repeats. More recently, in 2013, CRISPR-Cas9 was successfully used to edit the genome in eukaryotic cells, specifically in mouse and human cells. Following this breakthrough, gene editing was then performed in plant cells. In 2015, this technology was first used on human embryos with a genetic disease, but was ultimately unsuccessful because of off-target effects. The following year, a cancer patient's T cells were genetically modified as a new experimental form of cancer therapy. Today, CRISPR is still progressing rapidly in the field of genetic engineering as it's widely applicable and has a range of benefits.
First and foremost, CRISPR is known for its efficiency. A change in a specific gene in the early embryo affects a subsequently developed organism in a large scale. One major use for CRISPR is the prevention of genetic diseases through gene editing before embryo development. An example of this is sickle cell anemia, a disease with autosomal recessive inheritance. This is especially promising for genetic diseases that are difficult to treat and manage. Another benefit to using CRISPR is the ability to introduce mutations and modifications quickly onto model organisms such as mice, and for agricultural purposes, like increasing crop resistance to varying temperatures, or to disease. Because of how easy and efficient it is to alter the genomes of organisms, CRISPR has sped up developments in a wide range of fields.
CRISPR-Cas9 has progressed at an astonishing rate, and while it is a huge achievement for modern science, it wasn't until gene drive systems that controversy really heated up. A gene drive consists of 3 main elements: a gene encoding the Cas9 protein, a gene encoding a guide RNA to target the desired site, and flanking sequences which allow the cassette to insert at the specified site. Usually an allele has a 50% chance of being inherited, but with a gene drive it has a 90% chance due to its ability to copy itself throughout the genome. This means that within 15 to 20 generations, a few infected individuals could completely invade an entire population. This is great news for scientists wanting to wipe out problem species such as malaria-carrying mosquitoes, invasive species such as carp, cane toads, and other agricultural pests. By inserting a gene drive, we could alter or even eradicate these species. Just infect a few individuals with the cassette, release them back to the wild population, and let nature do the work. The decision of what to edit and why is multifaceted and should be openly discussed by scientists of various disciplines and the community, to ensure that if we do alter a species, it is not a short-sighted decision but has been carefully examined at all levels.